What inspired you to pursue a career in computational biology, and how has that inspiration or motivation changed or evolved over time?
So, that's sort of an interesting question. I think, I almost ended up here accidentally. And like a lot of bad decisions I've made in my life, the story begins with I had a girlfriend who, so my PhD was actually in theoretical physics. And I got my PhD in 1990. And um, it was a sort of interesting time because 1990 was, this may be ancient history for you, but it was at the end of the Cold War. Our biggest nemesis was the Soviet Union. The Soviet Union was coming apart. And so a lot of the justification for physics research spending in the US was because um, we needed to train lots of physicists because otherwise the Russians would have more, and that would be bad for national defense. So funding for physics started to collapse, and I actually had a job lined up in theoretical physics, and the support for it fell through, and so I had to do something else. So I started working with um, an experimental physics group at UCLA where I did my PhD, and that sort of fell through. So after 2 years, I needed something else to do. In the background, this idea developed to sequence the human genome. And it's really interesting. The Human Genome Project didn't start at the National Institutes of Health. It actually started at the Department of Energy, which used to fund physics research. And that sounds crazy, but why? And the answer is because the Department of Energy ran the big national labs. They figured that something the scale of sequencing the genome, right? Like the cancer moonshot today, solving these big problems would require big investments in national labs like big physics. And so they started this process of talking about doing something like the Human Genome Project. Now, it's not that crazy because when the Human Genome Project was starting, the Department of Energy was actually doing biological research. You think, well, why? And it's because its roots are in the Atomic Energy Commission, and part of their mandate in the DOE, Department of Energy, was to understand the effects of ionizing radiation, right? Genetics. So it was sort of genetics, big science. Eventually, the the NIH got involved. And so it was probably the right move since, it's really about understanding the effects of the genome on health and disease, and that's where it naturally sits. So I was doing my PhD in physics. I got my PhD. I did this extra 2 years working in experimental physics. And I had a girlfriend then, and I used to sit in her lab late at night because I was doing theoretical physics. It's a bunch of math, right? I could do it anywhere. So I'd hang out with her when she was doing experiments. And I discovered all this biology had been invented since my last high school biology class. So you probably know more now than I did in 1990 when I was moving into this. Um, but I would start helping her and her colleagues analyze data. And so I realized that if you had a little bit of mathematical skill and a tiny bit of knowledge, you could actually have an impact on research. So when my second physics career in experimental physics looked like it was going away, um, I impressed a number of people in her department, and they suggested it might make sense for me to think about doing genomic research. And there was a new program that had just come out from the National Institutes of Health that had taken over managing the Genome Project. And they were looking for mathematicians, computer scientists, engineers, chemical engineers, right? I mean, almost anybody with a quantitative background and skills to work on the Genome Project because they realized that this was going to be a big data project, right? The genome is 3 billion bases in length, the DNA bases with subunits. You have two copies of the genome in your cells, one copy is 3 billion bases long, right? That's a number that doesn't make much sense to me. It's slightly higher than my salary, tiny fraction of the national debt, but 3 billion is the number of seconds in 95 years. Okay. So that sort of
Yeah.
puts, a scale on it I can comprehend. And so we're talking about a lot of data. And so they realized they needed people with good quantitative skills. So they put out this call for proposals for a 5-year fellowship to work on the Human Genome Project. I was one of the first 3 people to apply. There were 2 computer scientists and some other goofy guy. And they gave out 2 awards, one to a computer scientist and one to the other guy. And that happened to be me. So when I wrote this grant, this application for this fellowship, I said I wanted to go into the lab and learn how data was generated, because if you're going to analyze data, you have to know where its warts are. You have to know what the challenges are. So I applied and ended up working with a group at the Salk when I got this award, working with a group at the Salk Institute in San Diego. I worked there for 2 years in the early days of the Genome Project, doing something called physical mapping. And sort of through a weird series of coincidences, I'd been there 3 months and I was in charge of the project, right? So I was doing that. The guy I was working with left, and I had the opportunity to move to Stanford. So I went from San Diego to the Bay Area, and I was working with a group there doing another kind of mapping, but they wanted to start a sequencing program because we were trying to put pieces of the genome together and then sequence them. And so we were moving to the point where we could start doing sequencing. So we thought, all right, let's do sequencing. And so we built the sequencing program. I worked there for 2 years. And in both these places, I combined laboratory techniques with building software and tools to make sense of data. And so for me, it was just kind of a natural thing with a quantitative background learning in biology. And after 2 years at Stanford, I had 1 year left in the fellowship, and I was thinking, okay, what do I do next? And at Stanford, there's sort of a glass ceiling. There's no place for me to go. I was a staff scientist, but I could never become a faculty member there. But I got a job offer from a place called the Institute for Genomic Research, which has its own sort of storied history in the Genome Project, which I can share with you if you're interested.
Yeah.
But I worked there, and I got offered a job. I moved there in 1997, and I quickly got my first large-scale research grant to try to decode what's in the genome. And so I can talk more about my research sort of from there. But that was really the thing that established me in this career, with this sort of progression through this fellowship, getting this research grant to begin to tease out what the genes do once we found them in the genome. And, I think you asked me what I like or what motivated me. I mean, part of it was that as a scientist, I'm basically a big dopey kid who likes to solve problems, and I like to solve hard problems. So one of the things that gets me very excited is the idea of tackling a big hard problem. And then a lot of what we've ended up doing is having to develop new methods that allow us to really take on these methods and take on these problems and make sense of them. So, the question I often get is, well, do you use what you learned in physics to do
Right.
computational biology? And the answer is yes and no, right? So I haven't solved a Dirac equation in more than 30 years when I was doing physics, right? But, I think one of the things physics does a really good job of is teaching people how to think and ask questions.
Mm.
And fundamentally, the most important thing about being a scientist is not the tools you take away from your degree, or not the tools you're even using day to day, but whether or not you can ask questions that help you get at the root of the problem you're trying to solve. So, for me, that opportunity to do science and to be this big dopey kid playing with some of the most interesting data imaginable and doing something people care about, right? People didn't care about the math I was doing alone in the dark room, right?
Right.
But people really care about, improving our understanding of health and disease. So that's been one piece of it. And the other piece that I think has just been so rewarding is to be able to work with really brilliant people and to build a team of researchers that pull together to solve problems. My research group is very interesting. It's I always tell people it's like a neo-socialist collective, that there's me and my students and my postdoctoral fellows, but a lot of times when they go on and get other jobs, they still come to our group meeting. I host them over Zoom and they participate. And eventually, over the years, they drift away, but they can always come back. If, somebody I haven't seen in 3 years, you show up at our meeting tomorrow and they be part of it. Because we built a network of people who trust each other, who work well together, who have this kind of mutual respect. And so it's really been, for me, a wonderful thing to be able to work with just amazingly talented people and learn as much from them as I hope they learn from me.
Right.
So I talk too much. If you want me to stop, just put up your hand or wave
No,
at me.
it's great. That was really interesting. Yeah, it sounds like a very interesting, I guess also unique path. I definitely resonate with the idea of problem solving as one of the reasons I'm interested in the field. What is an interesting fact or a fun fact kind of, or interesting thing that you would like to share, I guess, about your career?
About my career or about me? Um,
Either.
so uh, the joke is, there's sort of a joke among um, my research team here at Harvard that I only know two things. So um, one is when we get a set of genes, we want to figure out what we want to do. I tell them to do what we call a gene set enrichment analysis using Go biological process terms. Although one of my postdocs is convincing me we should use a pathway database instead of Go biological process. But this is one thing I learned. And at the heart of this is the idea that if we look at healthy cells and disease cells, and we find that in disease cells, a class of genes are turned on, what we want, or a set of genes are turned on, or others are turned off. One of the questions we really want to ask is what process is being activated by those genes being turned on, or what process is being shut down by them being turned off. And so whenever we get do these analyses and get a list of things, I always just say, well, tell me what it means. Do this kind of analysis. So that's one of the things. The other thing that I quote for people all the time, and this came from a PhD student I had years ago, Amira Jabari, who was actually a computer science student. And she showed me this paper, a 1997 paper by two computer scientists who were working at Microsoft, Wolpert and McCready. And the title of the paper is No Free Lunch Theorems for Optimization.
I've heard of it.
And yeah, I mean, it's a great paper. And there's lots of stuff in there that's interesting that I quote all the time. But the essence of it is this: if you're trying to solve a really hard optimization problem, the best way to solve that problem in finite time and get a near optimal solution is to start with a guess and then optimize around that guess. And the better the guess, the faster you get to a solution. So it kind of has flavors of Bayesian statistics where you use prior knowledge and optimize around those to get a good estimate of the significance of some finding. Or, it really has a lot to do with looking at a search space and trying to find, the mountain in a plateau. The closer you can get to guess to where the mountain is, the less you have to wander around until you find it, right? And so a big thread in a lot of the work that we've done over the years is just this idea that we can take some prior knowledge about the biological systems we want to understand and build that in kind of as a soft constraint, because our understanding isn't perfect, but it's a reasonable guess we can optimize around.
What is some of the work accomplishments that you're most proud of, and how would you describe them to a general high school audience?
So it's a great question. Yeah, I think in terms of my career, one of the things that I'm most proud of is that in 2022, I got elected to the National Academy of Medicine. Right. So that's something I'm really proud of. I'm very proud of the fact that, somehow being a failed physicist, I became a Harvard professor. Um, but in terms of the work we've done, I think it's been incremental. And if you ask me at different periods in my life, I pick different things. So I'll probably pick the most recent things because they're the things I'm really excited about and proud about. So in 2013, Kimby Glass, who's then a postdoctoral fellow with me, she's now about to be promoted to associate professor at Harvard Medical School. She is also by training. Kimby and I were working with some other people, and we reverse engineering gene regulatory networks. And it was really, my screen gave me a thumbs up, so it's very happy about this too. Um, and, and the idea is really sort of interesting. It's built on this no free lunch theorem idea that what we do is we model the process by which genes get turned on and off, right? So earlier I was talking about, gene expression and genes getting turned on or turned off and doing this gene set enrichment analysis. But what you want to imagine is that the Human Genome Project was a project to identify all of the genes in the genome. And we found about 25,000 genes. And one of the things that we got very excited about was, at least early on, a lot of people got excited about was, well, if you find the mutations in these genes, we're going to understand all disease. Which, on the face of it, is absolutely ridiculous, right? Because if you think about this for more than about 10 seconds, you realize the logical fallacy, right? Every cell in your body has more or less the same genome, right? So a brain cell and a liver cell are going to have the same DNA. What makes a brain cell different from a liver cell? It's not the DNA. It's in the context of that cell, whether or not you're turning on neurotransmitters in a brain cell or digestive enzymes in a liver cell, right? So that same DNA, which includes 25,000 genes, so it gives you a parts list of 25,000 things, it's put together differently. Regulatory process, right? So in brain cells, certain genes are being turned on, and liver cells, other genes are being turned on. And one of the things we understand is that for a gene to get turned on, right, we need basically a switch to be flipped, or to be turned off, a switch has to be flipped. And the way we envision this happening is, if this is a gene, to turn this gene on, I have a protein called a transcription factor that's going to come in, it's going to bind to the DNA. And when it does, it's going to turn that gene on and start making an RNA that makes the protein encoded by that gene. But the gene gets turned on when the transcription factor comes and binds. And with the genome complete, we can actually take each transcription factor, I dropped my transcription factor, we take each transcription factor, and we know enough about where they bind, the DNA sequences they bind to, that we can map them back to the genome. And not every gene gets bound by the same transcription factor, not every transcription factor binds every gene. So we can make a guess as to the transcription factor gene network, right? So we start with that guess, and then we take other pieces of information. If I have two genes and the same transcription factor is going to regulate both, I expect them to turn on together. If I have two transcription factors and they bind together, I'm going to imagine that they might work together to turn genes on. So we take these pieces of information, we all sort of synthesize it together. And what we do is we produce a model of how genes get turned on and off, a gene regulatory network. And I can produce a model for a brain cell and a liver cell, which is probably not that interesting. Or I can take a normal cell and a tumor cell, which is much more interesting, because now we're saying in the tumor, what's turning on? So this was a really foundational method. And when we published it in 2013, we named the method PANDA. And Kimmy can tell you like that what PANDA means as an acronym. I forget. N is networks, D is data, but beyond that, I forget. But it's a way of taking large genomic data and teasing out these regulatory networks. So that was really the first method. And since we've developed a whole host of other methods that have variations of what PANDA does, we have a slightly different method called Condor, like giant South American. We have Alpaca, which is the structure of the networks. We have Otter. I mean, there's a whole list, and we call this set of open source tools the Network Zoo. So if you look up Network Zoo, you can find our software and tools, and you can actually come in and use them. And there are a series of tutorials that Marwan Ben Gabila really was responsible for helping to launch. And these tutorials will teach you how to use the tools, right? So, I'm really proud of the fact we've developed these tools and been applying them so broadly. And I could talk about individual studies we've done. Some of them are very interesting. But one of the things that has really become an underlying theme in a lot of our research, and I credit my colleague Don DeMeo for pushing me in this direction. But one of the things we try to do is we try to understand why so many diseases manifest differently in males and females. And this is really interesting. As a male, I have twice, half the lifetime risk of developing Alzheimer's that my wife does. Okay? Given the fact that she's female. Even correcting for the fact that she's likely to live longer than I do, she's at much greater risk. Now, of course, genetic factors play a role, and we know there's a gene mutation called ApoE that increases your risk. But if she and I both carry the ApoE mutation, she would have twice the risk of developing Alzheimer's that I do, right? Why? My answer is it's got to be regulatory networks. So with Dawn and Camila Lopez Ramos, who's been a long time colleague and collaborator and is now starting her faculty career. Years ago, we published a really nice analysis of colon cancer. And we took tumors that had been removed by surgery before anybody got chemotherapy. And we built gene regulatory networks in males and females, and we compared them, right? So we're basically saying we have two networks. How are they wired differently in males and females? And if you throw away the things that are similar, more or less, and look at the pieces that are different, it turns out that males and females regulate a number of pathways, including drug metabolism and drug transport, differently. And when we saw this, we got really excited. You know why? Males respond better to chemotherapy than females do, right? So, why are females responding differently? Because they're wired to metabolize and transport drugs differently. And the really cool thing was when we took the females and divided them into those who had more male-like networks and those who had less male-like networks, the females whose networks looked male-like responded better to chemotherapy than the ones who looked less male. So this is just so exciting because it explains something we were never able to explain. It allowed us to start to think about ways in which you could intervene, right? So how do you make a non-responsive network look more like a responsive network? And it really required that we sort of ask and answer questions that pointed out flaws in our methodology. So Don and I actually wrote a grant to sort of patch some of the holes in the methods we used. But it's really, that project initiated a whole set of research now that's underway in my group that looks at differences in disease between the sexes. And then coming out of that work, actually something else really interesting, that some of these changes that we see between males and females are associated with aging. And aging plays a big role in disease too, right? Most, aside from some leukemias and one or two other neurological tumors, almost all cancer is a disease of aging, right? If I look at something, another disease we study is chronic obstructive pulmonary disease, COPD. As we get older, we all lose lung function, and people with COPD, it's accelerated. So one of the things we're starting to study now and dig into is just how the aging process gets altered in diseases like cancer. And again, what we want to think about is, are there strategies that we can adopt that are going to reduce the likelihood that we develop these diseases in the first place? So, it's really fun tackling these problems. And, I tell this story that the day when I was doing my PhD, I realized I had become a scientist was when I was doing this math and working on it for months and months and months. And I solved the problem and I was convinced that I was right. I was the only person in the world who knew the answer to this problem, right? I mean, think about it. You are now this singular individual in the world with a piece of knowledge. How valuable, it's up to you. But you know something nobody else knows, right? And then, of course, you share it. We have those moments in our careers so many times when you get to some point you think, I understand this thing. No one knows. And I'm going to share it with the world so somebody else can build on it and do more. And yeah, that's, I mean, that's been really exciting to me to solve these kinds of problems. And, I mentioned sex. We're talking, of course, about biological sex in this context. But Don and I are also very interested in trying to understand the interface between sex and gender to understand, for example, how much of this is chromosomal, how much of this is hormonal, how much of this is lifestyle. So, every time we ask a question, it's like Pandora's box opens. I guess Pandora's box is a bad example because all the horrible things came out except hope at the end. But, we're sort of opening up a box of, exploding glitter where all these things are coming out that you just want to grab and start asking questions about. And that's just been extraordinarily exciting.
What are some breakthroughs, I guess, in your field that you're looking forward to most that might come in the next 10 years?
So, it's hard to know, it's hard to predict what the breakthroughs are. A quote, and some people attribute it to Yogi Berra, a baseball player who has a lot of, bizarre quotes. Some people I think attribute it to Niels Bohr, the quantum scientist. Um, but the quote is, "Prediction is risky, especially in the future." So it's hard to know, where things are going. But I think somebody sitting in my position, what I'm most excited about, looking to the future, is just the idea that we're going to have access to data sets that will allow us to ask and answer questions we couldn't conceive. We may have conceived of asking, but we could never have conceived of answering. So, one of the things that I like to point out, and I use this is a quote from me, so you can quote me one day when I'm old, and I guess I'm old already, but when I'm famous. And one of the things I like to say is that every revolution in science has been driven by one thing, and that's access to data, right? If you have data, you have the raw materials to build a theory, to build a model. You have the raw materials to falsify that model if it's false, and then to start and build a new model. So I think new technologies are really going to rapidly accelerate our ability to ask questions by generating unprecedented types of data. Okay, so there's a plot I like to show, and it's the cost of sequencing a genome. And when the first genome was sequenced in 2000 or 2001, forgetting what it might have cost to sequence it, a pretty good estimate was the cost to sequence the second genome. Oops, I'm getting thumbs down. Cost of sequencing a second genome was 100,000 US, $100 million, right? Again, slightly more than my salary. But the cost started falling. And then in 2007, new technologies were introduced and the cost plummeted, right? It was dropping by about a factor of 2 every 18 months, kind of akin to Moore's law in computer science. But then in 2007, it started by dropping by a third every 3 months. Okay? So at some point, it almost made sense not to generate data, because if you waited a couple of months, you could generate a whole lot more. But by 2009, the cost had gone from $100 million to $100,000 to sequence a genome. Okay? So if I look at that, I would say this in 2009, if my wife or son had a rare tumor, I would mortgage our house and sequence the genome. Okay? Today, the cost is less than $1,000. The first genome took years. The next one took most of a year. 2009, you could probably do it in 2 weeks. Today, I can sequence your genome overnight for less than $1,000. Okay, so the cost of sequencing my genome costs less probably than your laptop, right? Unless you have a Chromebook, maybe then it's equivalent. But the cost of sequencing a genome, I can now pay for with a credit card. And making that statement just, I think, puts into context our ability to generate data. And so instead of having one or two genomes like we did in 2009, we have millions of genomes on health and disease. We also um, have access to new technologies.
Right.
So, one thing everybody's very excited about is single cell genomics. So, I used to take a chunk of tissue and I'd sequence that. But if I have a tumor, there are tumor cells and normal cells and white blood cells and all kinds of stuff in the tumor. Can't separate it out. But now I can disaggregate that and sequence single cells. And so now I can tell how many cells are white blood cells, how many cells are tumor cells, which fraction have which mutations, right? So suddenly I have a new microscope because I can look far deeper than I could just by taking a chunk of tissue. And so now we have the ability to do what we call spatial transcriptomics. So beyond taking single cells, now I can take a piece of tissue and I can put it down in a microscope slide and I can start to image in that tissue where individual genes are expressed. So I can know what cells are turning on what genes and how those cells are interacting with each other, right? Why is this so cool? Well, most of us over the course of our lifetimes are going to develop many tumors, but our immune system is just going to wipe them out, right? So part of what tumors often do is they evolve methods to escape the immune system. We can start to ask what those methods are, how they interact with the immune cells, how the immune cells interact with each other. And we can ask questions that were unthinkable 3 or 4 years ago, right? So we see this rapid advance in technology and it's giving us just unprecedented types of data. The cost is falling, so we can build bigger data sets. And then everybody gets excited about computation, right? Oh, AI is going to solve everything. I go back to the no free lunch theorem. If you take AI models and train them appropriately and constrain them in smart ways, then you can build models that are going to be predictive and explainable.
Mm-hmm.
So that's really exciting to me too. We just published a paper. I talked about Panda.
Yeah.
With Intikab Hussain and Jonas Fischer and Rebecca Burkholz. We published a paper in a method called Phoenix that allows us now to model gene regulatory networks over time. And so now we can start to ask lots of questions, but we were able to do it by me beating on INTA saying no free lunch theorem, no free lunch theorem, no free lunch theorem. Let's guess a network and optimize her. So, all of these things are exciting. And I think the other thing I would say to your audience is that what's so exciting is just, again, for me as a scientist, is as new people come into my group, they have new perspectives and they ask new questions. All right. And sometimes the answer is we can't address those questions because we don't have the data. But sometimes those questions make me stop and think, oh, we should have thought of that before, right? Like Dawn talking about sex differences. You should have thought of that before. And then once you do that, you just start building momentum in trying to come up with ways to address it, understand.
That's really interesting. And so, what advice would you have for young people looking to enter your field, and what specific maybe resources would you suggest?
So I think one of the most important things that I would tell people if they get interested in this field is it's really a field that exists at the interface. And the people who are, I think, probably most successful are those who can straddle the interface. And that's an interface between computation, technology, and biology, right? So for me, it's evolved to being a lot of computation, less technology. I rely on other people to invent the technology. But understanding the basic biology is really important, right? So, yeah, the great thing about statistics is that you can find things that are statistically significant. The terrible thing about statistics is you can take a big enough data set, you can find things that are statistically significant but absolutely meaningless in terms of the biology, right? So being able to use biology to frame questions and then interpret the results are really important. Having good computational skills, no matter what you do, if you want to do computational biology or be an accountant or invest in Wall Street or, run your own business, right? Understanding computation is going to be absolutely essential going into the future. So find a passion, something that excites you. If you want to learn biology, learn biology. But no matter what you do, learn basic computation. Statistical tools are fabulous. And if I had to tell somebody to learn a programming language, it would either be Python or R.
Mm.
Um,
Uh,
so
yeah.
Python is really big in the machine learning community. R is really big in the statistics community. Statisticians and computer scientists argue over what's better, machine learning or statistics. They're two sides of the same coin. They're techniques that allow us to get at relationships in data and outcomes.
Who are some role models or mentors kind of in the field and how they influenced you?
So, it's a really good question. When I started working in this field, there weren't that many people who had done it. Sort of the big field in computational biology at the time, the big advance was taking DNA or protein sequences and lining them up to each other. And there's some people who played really pivotal roles um, in developing those methods. So William Pearson is one, David Lippman is another. For aligning protein sequences, Margaret Dayhoff made just foundational um, contributions by figuring out that proteins or amino acids can't be evaluated just on identity, but they have to be evaluated on similarity and coming up with a robust way to do it. That was sort of the foundation then. Um, but in building this field, a lot of what we were doing, there wasn't somebody I could point to and say, I want to be like him. Um, but the the one person who I think, if you had to ask me of anyone, um, well, this gets really hard. If I think about my physics career and my biology career,
Yeah.
in physics, it wasn't my PhD advisor. It was actually a guy I worked with um, as a postdoctoral fellow in physics. And he's a very good friend and colleague. His name is Katsushi Arasaka. He's in the physics department at UCLA. He sort of now followed my lead and does all biological applications. But I was working with Katsushi, and the thing that I learned that was most valuable from him is just the importance of treating, of really learning from everyone. Like, my opinion mattered as much as Katsushi's did when we're trying to solve problems. And that was really encouraging. It let me ask questions and take risks. And I used to get off the elevator in the morning. Katsushi was the morning person. I would get off the elevator and I'd be walking down the hall and you could hear me. How? I don't know. And he'd come out and run and grab me and he'd say, idea. And he'd take me into his office. And he'd start writing things up on the board. And I'd look at it and look at it and look at it. I'd get to some point and say, no, 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 no. And I just walk out and sit there thinking, and then I'd think about it during the day and I'd come up with a way to solve the problem. And I'd walk back into his office and I'd say, look, and I start writing on the board. And at some point you say, no, 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 no. And it was just such an organic back and forth. And, to me, that was just so exciting, not to be doing science on my own, but to be doing as part of a team where
Mm.
everybody's respected. And that's an ethos I've tried to build. The other person who really had a big impact on me was somebody at Stanford, not somebody I worked with directly, but someone who became an informal advisor, and that's Ron Davis. And um, at one point, when I was thinking about leaving Stanford, I had a couple job offers. And I went to Ron and I said, look, here are my offers. What should I do? And Ron said, um, you know, you should go work at this place called Tiger, the Institute for Genomic Research. And there was a guy, Craig Venter, who ran Tiger. And I said, but Ron, you know, you don't have a lot of good things to say about Craig. Why should I go there? And he stopped and he looked at me and he gave me the best piece of career advice I ever got from anybody. He said, it will be a good place to be from. And it was really profound because Craig was a brilliant guy. He was horrible to work with, but he was a really smart guy and he gathered smart people around him. And so it was a really intellectually rich place to work because of my colleagues. Mm. And when I left, I established myself in the field. And then if I had to point to one other person as I got more senior, so I'm now chair of the biostatistics department at the Harvard School of Public Health. And in 2018, I was physically based at the Dana-Farber Cancer Institute with my academic appointment here at the School of Public Health. And I was getting frustrated with my ability to contribute to the organization as a non-physician. And so I got recruited for a job at another institution leading a big data science initiative. And I was starting to think about that. I was almost ready to take the job. And then the dean of the School of Public Health, Michelle Williams, stopped me and said, I don't want you to leave. Would you be interested in taking over as chair of your department? I thought, wow, you know, go and sort of build this data science institute or take responsibility for drop Claire a note. She phoned me. I was sitting in the parking lot at Home Depot. And she called me up and she started talking through my options. And, you know, she said, well, you know, what do you want? What do they want you to do? And I said, well, they have this dream of building this $100 million institute. How much money do they have? About $3 million, right? There's sort of a gap. And she had been asked at one point to go and build a big genomics institute at the University of Maryland. And she said, the biggest challenge with a job like that is that when you walk in, people are going to act as if you already have $100 million. So everyone's going to have very high expectations for what you're going to do for them. But without a lot of money, you're not going to be able to do all of these things, right? And so she said her first few years in building this institute were absolutely horrible because nobody was happy, right? But that may not be true, but you know, people weren't at least excited because they couldn't get everything, everything everywhere all at once. So she said, you know, how big is the budget at the School of Public Health? And I said, well, you know, our department has a huge research budget, right? And so she said, well, look, you can go into a place where there's a really strong research program already. And what you can do is you can help people do research better, right? You can create the infrastructure and the environment where people feel good about working together and collaborate and do new things together. And she said, you could do that and hit the ground running, or you go to a place where you're going to struggle for a few years. And, you know, I mean, Claire, it was just such good advice. She was a phenomenal scientist, but just the personal career advice was incredibly useful. And so finding people like that who can help you along the way, like Ron and like Claire, is almost as important as finding somebody that, you know, can help you figure out what the right career path is. Mm-hmm. So, you know, those three people were really fundamentally important to my career. None of them do anything related to what I do today, or at least didn't at the time. But it was just, you know, such enriching relationships with these people that really facilitated my personal growth as a scientist.
Thank you. I think that's about it for what I have. Is there anything else maybe that you would like to say or share?
So, you know, I think the other really important thing everybody should keep in mind, I always have too much, so two things, right? One is if you're going to do a job like this, you have to love it. If you don't love it, don't do it. Do something else. So, you know, for a lot of high school kids, they have pressure from their parents to do things, they have outside pressures to do things. If you don't love what you do, you're not going to be able to do it well. My son is 18. He's starting college in a few weeks. Um, when he was young, there was a hope that he would follow my footsteps, but he's going to University of Chicago to study environmental science and economics, right? He may change the world far more than I do, than I could. So, and he's excited about it. And I think that's really important that you have to find the thing that's going to make you excited. Because when you're coming into work at, 7 or 8 o'clock in the morning and you're going home and once your kids are in bed and your, spouses, watching TV, you want to sneak away and do more work to get caught up, right? It's a hard job. And so you have to love it. So if you want to do science, do it because you love it, right? The second thing I would say is don't be afraid to ask stupid questions. The great thing about being a physicist and working in biology was I wasn't expected to know anything, so I could ask stupid questions. And about 10% of the time, they're actually pretty good questions, right? And then as I built a research group, the ethos has always been if I say something is stupid, tell me, right? I don't know everything. I know I'm brilliant, right? Only 10% of the time. Help me find those 10% because I'm going to help you do the same. So don't be afraid to ask questions and be brave when you ask questions and push the envelope. And then, I guess the last thing I would say is that, you know, as you approach your career, don't be afraid of change. I was doing theoretical physics, I jumped to experimental physics. I was doing experimental physics, I jumped into biology. I was at the Salk Institute, I jumped to Stanford, jumped to the Institute for Genomic Research, jumped to Harvard, almost jumped out, stayed at Harvard. Um, and, you know, chance happens and weird things happen. And if you get knocked down, you just have to get back up. And in a boxing match, you know, the first guy who goes down and stays down loses. So if you get knocked down, you just have to get up and try again. And so, it's important to be adaptable, and it's really important to think strategically about what you're going to do and take risks and take chances. Um, and then I guess last but not least, build up a good set of peers and mentors who are going to encourage you and help you think in better ways about the problems you want to solve
Okay, thank you very much. Um,
and learn to program.
yeah, thank you. I've had a great time talking with you. Thank you so much again for meeting with me.
Oh, it's really a pleasure, Max. I'm so happy we had the chance to talk. My
Me
last
too.
name is Quackenbush, and so if you look me up in Google, John Quackenbush, I'm easy to find.
Okay.
If people see this and want to drop me a note, um, please feel free. I can't promise I'll respond immediately. I may miss your email, so if I miss, write back. But as you can probably tell, I love what I do and I love talking about it. Maybe too much, um, but if people have questions and want to talk more, please feel free to reach out.